Okay, well, very good afternoon, our respected resource person, Dr. THIRUNAHARI UGANDHAR sir, Associate Professor, Department of Botany, Kakatiya Government College, Autonomous, Hanamakonda. Sir is going to explain about the most important topic, that is cytotaxonomy. It is very important. It deals with the cytological aspects in the taxonomy for the classification of the plants. So it is very, very important topic, my dear friends. So listen carefully. Sara has huge experience in the field of plant sciences, plant molecular biology, biotechnology, tissue culture, and also the molecular taxonomy. So that's why it's a most important topic. Sir, please start your session. Hearty welcome, sir. Please unmute yourself and start your session, sir. Welcome, sir.
Yeah, happy birthday, sir.
Thank you very much, sir. So really, I'm very thankful to you, sir, because of your posted in the Facebook. So many friends are wishes me regarding of my birthday. So thank you very much, sir, for your warm blessings and wishes. Because of your the very senior most botany fraternity, your wishes definitely promotes me in progressive way. Thank you very much. I need your blessings and suggestions and also the guidance. Thank you very much, sir. So please start your session, sir.
Sir, right. So then today we are a cytotaxonomy. Cyto means also cell. Taxonomy means also identification, nomenclature, and classification. The relationship between the cytology and taxonomy is also cytotaxonomy. It is a more interested topic. Then it can be clearly explain the cytotaxonomy. So what are the meaning of the cytotaxonomy? The cytotaxonomy is a branch of taxonomy that classifies studies organism based on the characteristic features of their cellular structure, particularly chromosomes. So it plays an important role in understanding the evolutionary relationship among the different species of plants. The evolutionary relationship based on the chromosome, the cytotaxonomic studies, the chromosomal features are considered a valuable tool for plants and animal classification, both. But here among these chromosome number, structure, size, chromosomal abnormalities, behaviors are the more major aspect to examined in the cytotaxonomy to related to the plant species in the same family or different family. So the chromosome number is the most widely used, the important parameter to the identification of cytotaxonomy. It helps in identifying species and also called detecting polyploid, monoploid, uploid, diploid. These are number that tracing evolutionary lineage. So the chromosome structure is also second important. So especially it can position of the centromere. Based on the centromeres, the chromosomes are metacentric, submetacentric, acrocentric, telocentric. The four shapes are different. That is also study structural rearrangements like a inversion and translocation is also important. Then duplication are also called transcriptions. Also, this is also chromosomal structure. Then chromosome behavior. It is also during the division of the mitosis or meiosis, produced to the insight into the particular segregation and evaluation of the compatibility or into the study of a genetical relationship. So it is. So thus the cytotaxonomy is a bridge cytology and taxonomy bridges. It is also both are connected to the cytology and also called a taxonomy. So both are relations. It is a majorly only cellular organization like a and chromosomes only. So it is the taxonomy offering the deeper insightness into the evolutionary and genetic connections between the organism, genetic connections. So then, what is a definition of the cytotaxonomy? The cytotaxonomy is a branch of taxonomy that focuses on the classification of organism through the comparative study of chromosomes. Only chromosome is a major part for relation to the taxonomy. It can particularly during the cell division of metaphase. During the metaphase, the chromosome structure is clearly seen under the microscope. That structure based to identification of the related species of same family or different families. So then, so these are the comparative study of chromosome, particularly during the cell division of metaphase. That is important. Cell division only metaphase. These approaches are instruction to the resolving the complex of the taxonomic issues. The fundamental principles of taxonomy is the closely related species found in the have a similar chromosome structure in the same species or different species, then collectively known as the karyotype. Karyo means that is also chromosome arrangement or ideograms, karyograms. It is also one. By comparing the number, size, shape, and structure of chromosome, the scientist can enter also evolutionary relationships and genetic similarities among the species. So the integrating of cytology with taxonomy as significant contribute understanding the evolutionary history of various groups of the plants in the same family or different family, in the same species of the plants or different species of the plants, including also fishes and also primates and many plant species also. The fishes or animals is also basic relation of the cytology in relation to the taxonomy. So the role of the unique strength of the cytotaxonomy is able to detect minutes chromosomal variations. The chromosome, these appreciations are makes to the powerful tool and of the backbone of modern taxonomy. Backbone of the modern taxonomy is called a cytology. Then before embryology. The embryology is also important, but not more than cytology. Means the cytology is an internal cellular organelle structure base. The chromosome identifications, the chromosome arrangement, the chromosome size, shape, and structure. It is a major important role to the classification of APG system in present days or current system of classification. The helping scientists to explore and define the natural relationship among the organism with greater accuracy. Then contribution of the cytology to the taxonomy. The cytology, the branch that deals with the structure and function of cell. Cytological characters play an important role in modern system of plant classification. That is also modern important means current system of classification. Many taxonomist problems have been solved using cytological chromosome data. The very and the non-current and non-evidential rules and evidential rules. It can based the two groups of scientists related or divides. That is also then some of the scientists are non-evidency and evidency. So these are total evidence. It can be related that can base of the cell structures. Then the chromosome data is important for relation to the evidential scientist and non-evidential. Use of cytology, the study of taxonomy is called cytotaxonomy. It can study of cytotaxonomy involves the study of karyotypes and ideograms of the species in question. So karyotype is a karyotype, what is also called ideograms. The importance of the cytotaxonomy, it can be crucial in solving taxonomic challenges. So cytotaxonomy plays a vital role in modern taxonomic studies, particularly resolving the complex classifications. It can use of the chromosome as a taxonomic character. First one, the cytotaxonomy law major significant role is called a use of chromosomes as taxonomic characters. So the chromosome have a long been also called and as a taxonomy tools. However, noted the Jackson 1984. So also despite the chromosome have been extensive cytological research, much of the potential uh, information also called remain undefinitionally also called taxonomist with improve the techniques taxonomy or cytotaxonomy more accurate and informative with the results. The taxonomy means only not the taxonomical relation to the cytology means that is cytotaxonomy. It details of the minerals variations. One of the key of advantage cytotaxonomy and physiological taxonomy. It's a ability to detect the suitable chromosomal differentiations. Ability cytology and also got a physiology means the physiology means the inner studies of metabolic activities based on the chromosome or genetical inheritance characters. Then cytology is the term of the karyotype is used for the phenotype appearance of the somatic chromosomes. The diagrammatic representation of the karyotype is termed as ideogram. Then the characteristic of the chromosomes which have proved the taxonomic values include chromosome number, chromosome size, chromosome morphology, and chromosome behavior during the meiosis. It is also a major part for the study of the cytological information and also called a cytological studies in the plant systematics. The chromosome number, first we did how many number of their chromosome number, size, morphology, and also got a behavior of the meiosis during the meiosis or chromosome behavior. But out of the four, you can take and let me know the chromosome number. The chromosome number fixed all individuals of a particular species, which is different from other species. If the chromosome number is the same, but the nucleotide arrangement is also different. That is care. The constant chromosome number of their species are taxonomic importance. So then change in a chromosome number occurs unapplied. Additionally, or deletion of one or two chromosomes, but not a complete set. Unapplied, also triploid or monoploid. So which is also called a polyploid means addition of one, two or more sets of the chromosomes. This can form a basis of cytotaxonomic considerations. So such studies have been used in the studying of taxonomy of some genera like Carex, Datura, Pteroxen, and also Chlorophytum. This is also major studies of the chromosome number or unapplied, polyploid, euploid, monoploid. What are the characters? What are the relation of these plant species? So, the tribes of the family composite have been also delimited the basis of the chromosome number. That so this is also the cytotaxonomical studies of a racine fishes of a Kashmir Himalayas, telos are sapronide, October 2011 carried fishes are 64/4. Means the karyotype study of a Shaijo rat species, which Shaijo niger, Shaijo ecosinus, then also called a Shaijo thorasin and Labian. Different species are there. Obtained from the Dal have been Kashmir was carried out the following methods. It is also first one is also yes, Labial 2N is 98. Then also Niger 2N is 98. Yes, is different in species of the plants, but different in species of the fishes. also same type of the chromosome. That is also the S. plagiostomus, 2n is equal 96. Then S. curviform, 2n is equal 94. Means generic name same, species names are different. The species name is also denoted to the number of the chromosome. It is also one approaches the chromosome size. The challenges in the size and structure of the chromosome can occur by addition of or deletion. Addition or deletion or rearrangement are also called a transcription. This is also important of the chromosome size. Few chromosomes are deleted. Few chromosomes are added. Additions also called a transcripted. This is also major qualities, major characters of the chromosome based on the size. These changes in chromosome size and structure are used. Cytotaxonomy is a rearrangement of taxa at various levels. Means the chromosomal behavior base same group of the plant species are separated in the phylogenetic classification. That is called a major event based on age importance or significance. Then chromosome behavior, the pairing and separation behavior of the chromosome during cell division helps in a taxonomic studies. The chromosome behavior is responsible for the development of aneuploids and polyploids and also origin of the new species. So this is also major important of the behavior of the chromosome during the meiosis. It can be major significant role in phylogenetic classification. Phylogenetic that classification. So which is also the karyotype of a Solanum niger and Solanum cruciferum. Also the different size. The size of the chromosome is also various, also different in structures. So this is a one type. Then the chromosome morphology. This position, centromere determine the chromosome morphology. Metacentric, submetacentric, acrocentric, telocentric. The four type of the different species or arrangements or different chromosome structures. It can major use to identification of the plant species. If different, same and different chromosomal characters having the plant species are in different plants, different families, that plants are separated to include into the same family by APG system, significant role. So the terminal segment of the chromosome present above the secondary construction called the satellite. So this is so the tribes of the family. So renonucleaceae have been regrouped based on the chromosome morphology. It is also called the the renonucleaceae families are majorly grouped into the non-phylogenetic. During the phylogenetic group, that is also rearrangements. It is also based on the chromosome morphology. The chromosome bonds. It is also one phylogenetic characters. The chromosome bonds are referred to the alternate dark and light regions. Dark and light regions along the length of the chromosomes. Means that is called a heterochromatin or euchromatin. It is also called a dark in colors of the total dark in colors of the chromosome patches is present. It can produce after staining with a dye, G bonding, R bonding, etc. So what is the G means? The staining with so GSMA, dark bands, AT rich and also light bands, GC. AT means adenine, thymine, and guanine, cytosine rich. That is also one character. R bonding means also called staining with geonema or geosma, dark bonds, GC, guanine, cytosine rich, and also adenine, thymine, also light rich. That is character. That characters base to the separated families in old classified group of the families. Then Q bonding, the staining which quinacrine, dark bonds 80, rich light bonds GC. It is also one characters. Then C bonding, the staining with chiasma, dark bonds contains a constitutive heterochromatin part. The heterochromatin part is also there in C bonding place, which is major important role to the refers to the chromosome and in dark color, also light color after staining that plant species are separated into the old family to new family based on the APG role. APG significant role or concept. So this the chromosome shape on the basis of position, metacentric, submetacentric, acrocentric, telocentric, subtelocentric. This is also several types. So then the centromeres, central in position, P and Q arms are equal in length. So example, so it is a pairs in a Solana Niger and also 15 pairs in a Solano Isudium or Solana. And so it is also one type of the fish or in animals structures are based to the separation of the chromosome structures. Acronymatic in the same way, arm very long, another very short. The telocentric is also centromere terminal in position, showing only one arm. Then subtelocentric means the centromere is closer to the end, which is also based on the animals of the fish structure. And the animal taxonomy also called a plant taxonomy. Both are complete depends on the cytology based on the internal or cytological behavior character. It can phylogeny relations. Then these are chromosome types. The chromosome and the heterocentric and hygrocentric. So one character system of cytological values. Then, so these cytological values of the studies, then Sharma 1950s, on the basis of studies of Araceae, Amylidaceae, and Diascoraceae families proposed that changes in karyotypes of somatic tissues play a distinct role in evolution. That is also, it is also a plant character. Then Araceae, Amylidaceae, and also guarded Diascoraceae. These three families are based on the karyotypic nature in somatic cells, tissues to separate the plants in same group, old group of the families. On the basis of these studies, proposed that large chromosomes, low chromosome number, or less chromosome number, and symmetric karyotype to represent two status, whereas small chromosomes are high chromosome number and asymmetric karyotypes also indicate the advanced stage. So this is the taxonomy of family. So Alismidaceae has also been studied using these Alismidaceae. Then so exemplified bread, wheat, triticum, aestium, 2n is equal to 42. Also got a durum, wheat, triticum, durum, 2n is equal to d8. And also got implied triticum, monococum, 2n is equal to 14. Can be differentiated in the base constitutions. Constitutions are, it is also
మనం బయటవాడు మనం పదనవాడు కాదు వాడు క్లియర్ గా వాడు కొట్టనే వాట తిట్టనే ఇంకా పదనవాడు ఏంది చక్కగా దానికి ఇచ్చుడని మాట్లాడే మాట్లాడకపోతే మనం నాన్న ఇప్పుడు అట్లా సైలెంట్ ఉంటాడు సపరేట్ ఇస్తాడు అవ్వకపోతే
also got a primates A.D. Stark, M.K. Miller, by being compared to the then also called a, so several number it can be
సార్ మీ వాయిస్ వినబడుతుంది సార్.
Okay, sir. Then when these two chromosomes are
yes sir.
when are fused together near the centromere of one chromosome and telomeres of another chromosomes. This is also chromosome evolutions of the primitives or character features. The system of values of cytological studies Johnson in 1971, the members of Cyperaceae and Juncaceae passes a chromosome with diffuse or non-localized centromeres. Non-localized centromeres with diffuse and also now inverted meiosis. So this reflects close association between these two families. Then which families? Cyperaceae and Juncaceae. It can pass as two relative families. So based on the chromosome or meiosis nature. So Yucca had long been treated as a member of the Liliaceae because of superior ovary and agave of Amylidaceae because of inferior ovary of Hitchens and shifted both players to agaveaceae because of the presence of 25 small and 5 large chromosomes of both of them. So then the Hitchinson scientist was based on these characters, it can shifted to the two families into then the systematic value of cytological studies. The basic chromosome number in Loranthaceae is N is equal 9. 9 number of chromosomes, while is a Viscaceae, there is a series of unapplied chromosome number rearranged are ranging to the 10 and 14 veins. 1975 separated them from each other and basis of cytological evidence. So these two type of the chromosomal group of the plants are separate to each other. That is chromosomal evidential character. In the subfamily, so Bambusudaceae of a graminease in 12 and the subfamily Podiaceae is 7. This indicates that the chromosome numbers have proved to the taxonomic utility also at the subfamily levels. Then first family is also gramineaceae, N is equal to 12. Second one, Poidaceae is also N is equal to 7. What is the difference? It have been proved to the taxonomic utility. It can be separated the two subfamilies. Then Stebbins, 1958, provided information on the evolution of a grasses. Evaluation of the grasses on the basis of cytogenetics. So it is also one character features to the identification of the plants. But the systematical study on the basis of cytological studies, Lewis, 1951, subjected the genus also Goritia in Carchia Onogris. Then also called a second scientist, Naik, 1977, differentiate the species of chlorophytum of Liliaceae on the basis of cytological data. Then according to the him, so chlorophytum, so Baruch as 2n is equal 16, while chlorophytum glaucum and chlorophytum glaucoides have 2n is equal 42. The chromosome number is also different in same genera, different species. Both the latter species have different karyomorphology. Then Warburg, 1938, studied taxonomy of Germinalis, Geranialis on the basis of cytological studies. This is also then also called a Mander, 1932, genus, and also it can be cistus or cistaceae formerly included in the Helianthemum as a chromosome number is 8, while Helianthemum is based on the chromosome number 9. It can be shifted to these two families. A new classification of the genus Narcosus of the Amylidaceae has been proposed by Finadaceae, 1951, on the basis of the cytological studies. It is also then, so these are all is also examples of the cytotaxonomy base. So what is a main what is the phenomena of the cytological relation in plant taxonomy means the chromosome number, chromosome size, chromosome shape, chromosome behavior, the four important for phylogenetic relations to the taxonomy. That is also these relations in the taxonomy, it can separate it to separate branch. That is called a cytotaxonomy. Then after next, which chemicals are made up of the cell? What are the secondary metabolites, character features? It is based on the chemicals. Then the based on the chemicals to classification of the phase into the chemotaxonomy. But here is also chemotaxonomy. Chemo means as chemical. Also called as means it is also inorganic components. Major metabolites are it is a separate group of families. That is also chemotaxonomical role. Then chemotax is the chemical sense of on the molecular characteristics. Different. Then also called the plant species which have the chemicals structure is can be product of a two quite different pathways. The cousin is called a chemotaxonomy. Chemical identification, chemical examination in the leaf, in the shoot, root, or fruit, seed, which is a
సార్ స్ట్రక్ అయింది సార్ ఇది